Pittsburgh East, we are very delighted to give you some updates of our progress. And also, we are very grateful with the funding support, of course, from NIDCR, and also for this retreat, we received funding from California Institute for Regenerative Medicine. So this is really fantastic to have this kind of support. From the very beginning, when we set out to do this dental, oral, and craniofacial tissue regeneration, we developed an overall vision for the C-Doctor program to become a sustainable and comprehensive national resource center that will help to enable the clinical translation of basic science discovery into clinical practice. And with the development of technologies that help to regenerate dental, oral, and craniofacial tissue and organs lost due to congenital birth defects, traumatic injury, or some other medical procedures. And we are currently in stage 3 of our DocTRC funding support. And when we put together this proposal for our stage 3 support, we outlined 4 specific aims. And I'll give you some updates on what we have been the progress we have made under each specific aim. And that will really help to set the stage later on when you hear the ITP presentation. So aim 1, we really wanted to coordinate and integrate the C-Doctor infrastructure and strategic partnership to support our interdisciplinary translational projects. So these are projects selected, some of them from stage 2, and then transitioned to stage 3 because of the terrific progress they have made. And then in this stage 3 funding support, we have put together this very customized team of support that will provide guidance on regulatory science, on how to commercialize product, and the GMP manufacturing, packaging, sterilization of products, clinical trial design, how to write a clinical protocol, and how to interact with FDA, and also project management. In this process, we also continue to interact with much broader external organizations, such as CIRM, the FDA. You know, we've had FDA presence at our previous retreat. You know, the discussion between FDA and also our ITP teams are just really productive in terms of learning how this process is supposed to be carried out when you file IND or IDE with the FDA. And furthermore, yes, you know, tissue regeneration has been brought up by many other institutes at NIH. And so we're in the process really interacting with different areas of NIH support to see how we can model this and continue to expand the impact we have from this program. So aim 2 is to basically assemble a very balanced ITP portfolio. These are all really based on our initial assessment, based on the clinical needs. What are the things we need in dental, oral, and craniofacial regeneration area that patients can use? And based on that, we selected ITP teams and helped them to make progress towards product development and clinical adoption. And currently, we have 9 ITP teams. 7 are in stage 3, and 2 are the new ITP teams with the pilot funding. And these ITP teams have set specific milestones as a part of the sort of evaluation process. And we have quarterly updates to look at the progress. And then this really, you will see from the ITP presentation later, they have gained a lot of interest from industry, from venture capitals and other investors. So this is a summary of the 9 ITP teams we're currently supporting based on the study topic, the PI, and also the tissue type, and also the type, whether it's a device or drug or a combination of both. So to better sort of give you a summary of what we have under our portfolio, here you can see on the left, these are the different technology types. So we have some projects that are based mainly with drug or device or a combination of both. In terms of the tissue type we have addressed, this really, I would say, perfectly aligned with our initial assessment. What are the tissues that we need to regenerate, such as bone, fat, skin, muscles, salivary gland, periodontal tissue, and nerve. In terms of the FDA interaction, which is really the goal of this DocTRC support, is to help to these projects from the basic discovery all the way to have a successful FDA IND or IDE filing. So more than half of our projects have had FDA interactions. And then more importantly, earlier this year, our the very first project out of both resource centers, this project led by Derek Wan on deferoxamine patch for radiation-induced skin project was received approval from the FDA. And the project led by Jeff Gardner, the scar formation prevention ITP, we also filed their IND and now in the process of addressing some of the issues or some of the studies that FDA requested. And we also have 2 other projects had a pre-submission, and one on the calvareal bone regeneration. And currently, we're addressing some of the feedbacks from the FDA, and I will go over that in detail later. And also the salivary gland regeneration led by Sarah Knox and Chelsea Bainey also had their IND submission meeting with FDA. And finally, the project led by Kevin Healy. So altogether, these 5 projects have had 8 FDA meetings, and they're really very active in this process. In terms of the other accomplishments, we have gained additional funding from other sources other than the NIH funding, altogether more than $20 million. And so with 22 patents issued over the course of our support, and then more than 40 publications. A lot of these are really highly impactful studies and setting the sort of the trend for the future study. And in AIM-3, we set to validate key ITP results and perform preclinical testing and developing a shared knowledge base for the standard operating procedure, protocols, and best practice. So what does that mean? So I'll give you some couple of examples. So the ITP teams, we really rely on each other, sort of a feedback. What are the large animal models that we should use for this particular project? So this has been one of the areas where we have had a lot of interaction and then sort of guiding the ITP teams. And other ITP teams discuss common challenges they encountered during like their study design or FDA filing. For example, the experience from the scar formation prevention, the ITP led by Jeff Gardner, their work helped to inform Derek Wan's project, and then that accelerated their FDA filing, and then resulted in a successful IND. And then also this has helped to inform the project led by Mike Longacre. And our experience switching for our Calvario bone regeneration project, initially using NeuroQuest-derived cells, now using bone marrow aspirate, has really helped to inform the project led by Tara Agalu on periimplantitis bone regeneration. So these are just some really examples that you can see how these kind of a network of experts have helped to bring the experts together to support our ITP teams. Finally, for AIM4, and because NIDCR has supported these two resource centers, and these two resource centers have been truly operating as one. We have frequent communications, joint decision-making. We serve on each other's boards as voting members, and Dave and Masumi are here from the Michigan-Pittsburgh Rees Group, and we have our joint annual retreat that's scheduled for October this year. And that's going to be held in Bethesda, where we're going to have more FDA interactions and also interaction with other institutes in NIH. So we're really also very dedicated to educate and train the future leaders in dental, oral, and craniofacial tissue or regenerative medicine. And also we have been putting together the two resource centers, putting together these webinars and recordings, and then sort of training sessions to just train people how to do regenerative medicine. So here I'll give you some highlights of our C-Doctor this sort of a year in review. The Hydronovel, the salivary gland regeneration project, ITP, led by Sarah Knox and Chelsea Bainey. They had their pre-IND submission, and also they were awarded this $2.3 million of CIRM trans grant, and very exciting. And as I mentioned earlier, Derek One's project received their IND approval, and the scar formation prevention led by Jeff Gardner now have funding from AFIRM. And so this is really very timely for, to support of their clinical trial study. And also our Calvario bone regeneration project, I will tell you a little bit more about our pre-IDE meeting with FDA, and also funding support from the Alfred Mann Institute. The cleft lip revision drug led by Mike Longacre, they are in the process of preparing for their pre-IND meeting. And the periimplantitis bone regeneration study led by Tara Agalu continue to have their discussion about forming a startup. And also the drug-induced periodontal regeneration. So they're led by Dr. Messer-Smith and Dr. Haber-Katz. They're in the process of doing their patent application and IP protection. And the craniofacial muscle regeneration led by Kevin Healy. They have just recently started this startup company called Muscle Matrix. And also they're beginning their commercialization activities. And finally, the project led by J.P. Pepper, the nerve regeneration project, they are in the process of discussing with Curis regarding in vivo use of this SAG21K molecule. So the DocTRC program supported by NIDCR as the two resource centers have really hosted multiple seminars. Recently, we had a seminar led by Charles Hart and also Gary Hammer, really looking at how disseminating the information of how to conduct translational research. And from that, actually, Charles Hart gave a talk last Friday at USC to really highlight how, to carry out these translational studies. So the impact is really being spread everywhere. And the DocTRC program continues to really receiving the funding support from CERN for our retreat, the last one, the joint retreat in LA, and then also this one here at Berkeley. And also we were featured in the NIDCR 75th anniversary celebration with our tissue regeneration projects. So looking ahead, we are entering our last year of our stage 3 funding support. What we have learned has been tremendous. And then more importantly, at this meeting, you will evaluate what ITP's project, what they have been, the progress has been made. But more importantly, we want to see what we can do together to expand this effort and use this network of experts to continue to support tissue regeneration efforts. I think the most important part, as you will see, is this very rich network of experts and consultants we have put together. And we'll continue to support our current ITP. And then our goalpost is very clear. It is to help these ITPs to have a successful IND and IDE filing, period. That's what we're working on, but also at the same time, we are very excited to see this new funding opportunity from NIDCR accelerating product excellence in innovation and for clinical adoption as this APEX program. So with this program, we wanted to align our effort with how to improve mentorship and training, career development for clinical translation of these basic research projects. And then also developing the process to apply or extend our model beyond the dental, oral, and craniofacial. So at this meeting, you will hear talks on wearable device and how do we use that to improve or monitor health. So this is something, this is perfect timing for this APEX program. And also building upon the relationship with other programs. NIDCR supported like TMD Impact, and we recently had a retreat on that. And this is about temporomandibular joint disorder, and also the interaction with other funding agencies, and industries, and institutes beyond NIDCR. And we wanted to continue to have that interaction. So these are the programs for the next 2 days. So today we're gonna start with the ITP presentation. And then once we finish that, we're gonna have a group photo right before lunch outside. And then after lunch, we are very delighted to have Lisa Kidak, who had been with us multiple times at this retreat, really talk about the California Institute of Regenerative Medicine's effort in supporting tissue regeneration. And then after that, we'll have a discussion on sensors and wearable diagnostic session. So that will be, the talks will be given by Jeff Gardner and also Uttam Sinha. So Jeff and Uttam are both here. And then after the break, we'll have our session on best practice. And then that will wrap up. So that's will be a session on the FDA interaction. So that'll be led by Dr. McCain and also Dr. Nan Kumar. I think Dr. McCain's here in person and Dr. Nan Kumar will join us online. So then we'll have dinner and then group interaction. And tomorrow we're gonna basically start the morning with bridging academic and regulatory science centers, and that will be led by Yunji Pacifici from USC, and also Kathy Giacomini from UCSF, who's the dean of UCSF School of Pharmacy. And then after that, we're gonna have a talk on lean Langevin talk about from the, who's the director of the National Center for Complementary and Integrative Health, and then talk about the potential interaction between our DocTRC program and then this other, our NCCIH Institute. Then we're going to have lunch, and afterwards we'll have our closed door sessions. So if you are joining us online, please mute your microphone. And also the ITP progress evaluators, please check your email for the link, and then that's where you enter your evaluation after each presentation. If you have any technical questions, please contact Vivi, Nicole, Pui, or Tom Sullivan. And we want to really take this time to thank our external advisory board, Marie City and Erica DeBauer, who couldn't be here because of some family emergency, Brandon Lee, and also Rob Sparrow, and also our NIDCR leadership. And I think Scott has some problem with travel and joining us online, and Lillian and others. Also the CERM and other people who are here, speakers and guests, and our CDoctor leadership, and also the operating team. Thank you very